Hello, my name is Jennifer Mydosh, and I am a graduate student in Dr. Carrie Cooper's Food Safety Laboratory. I'm going to tell you today about the phenotypic and genotypic comparisons of Campylobacter jejuni strains with different clinical outcomes. To give you some background, Campylobacter jejuni is the leading cause of bacterial gastroenteritis worldwide. Infections typically present with either a watery diarrhea or a bloody inflammatory diarrhea. However, little is known about why there are these clinical differences. My research aims to answer this question. Comparative genomics were done on 4 Campylobacter jejuni strains associated with watery diarrhea and 5 Campylobacter jejuni strains associated with bloody diarrhea. The core and accessory genes were assessed in each of these strains. Phenotypic assays were done on 10 strains of Campylobacter jejuni, 5 that cause bloody diarrhea, and 5 that cause watery diarrhea. These assays include motility, biofilm formation, CDT production, attachment to intestinal epithelial cells, invasion of intestinal epithelial cells, as well as generation times. In table 1 here, you can see a table that will give you some more information about the 10 strains that were worked with. I want to note that strain A13A is currently being sequenced by the Cooper Laboratory, so it is not included in the genomic comparison. However, it is included in all of the phenotypic assays. Figure 1 here can give you a good illustration of how distinct these differences are. These are the intestines of neonatal piglets that were challenged with either a strain of Campylobacter jejuni that causes watery diarrhea or a strain of Campylobacter jejuni that causes bloody inflammatory diarrhea. Figure 2 is a phylogenetic tree showing how the different strains are related to each other. You can see that the strains are not grouped by a clinical outcome, and strains that are associated with inflammatory diarrhea are closely related to strains that are associated with watery diarrhea. Figure 3 is a blast atlas that is showing the genomic comparison of C. jejuni strains compared to strain RM1221, which is the strain that has the largest genome, and compared to the other groups of Campylobacter jejuni. So the green is the reference genome, and the light blue are the strains that are associated with bloody diarrhea, and the light green teal color are the strains that are associated with watery diarrhea. The white spaces are the genes that are absent from their genome compared to the reference genome. And from this figure, we can see that strains associated with bloody diarrhea have the most absent genes. However, there are no genes that are associated with either clinical outcome. Moving now to the phenotypic assays. For these assays, for these figures, the strains that are associated with watery diarrhea will be colored light blue, and the strains that are associated with bloody diarrhea will be colored a light red. So in figure 4, we have the attachment assays done with INT407 and cacao2 BBE cells, which are both intestinal epithelial cells. And there is a statistically significant difference between the attachment of strains associated with bloody diarrhea and strains associated with water diarrhea in the INT407 cells, but there's no difference between attachment in cacao2 BBE cells. Figure 5 is biofilm production, and figure 6 is motility, and there is no statistical significance between the groups for both of these assays. Below, we have the invasion assays, and for this, we do have a statistically significant difference in invasion with the strains associated with bloody diarrhea for both INT407 and cacao2 BBE cells. The strains associated with bloody diarrhea invade much more than the strains associated with watery diarrhea. Finally, we have generation time and CDT toxin production. So strains associated with watery diarrhea are statistically significantly faster growers than strains associated with bloody diarrhea. And strains associated with watery diarrhea produce more CDT than strains associated with bloody diarrhea. So in conclusion, we found no genes that were associated with either clinical outcome. However, there were 18 single nucleotide polymorphisms, 12 of which were associated with a particular outcome. And we also found that there were SNPs in the critical two-component regulatory system, RAC-RS, only with strains associated with bloody inflammatory diarrhea. The phenotypic assays revealed statistically significant differences in strains associated with watery diarrhea for CDT production and generation time, as well as strains associated with bloody diarrhea are able to invade much more. This study provides us with the foundational knowledge to pursue future studies looking at the role RAC-RS plays in the differential clinical manifestations, as well as further investigations to solve the differential pathogenesis of Campylobacter jejuni strains associated with different clinical outcomes. Thank you very much for your time.